In my previous module, I talked about activation energy and also the speed of energy. Let's talk about that more. Delta G, change in free energy, does not tell us about the speed of a reaction, how quickly a reaction will proceed, how quickly the reactants will be converted into products. Delta G just tells us about the equilibrium point of the reaction. So enzymes are the entities that can manipulate the activation energy. That's the topic of this module. We will see how enzymes can manipulate the activation energy. To understand activation energy, we can go back to our original analogy of ball rolling down the hill. In this case also, our products are at higher energy level, reactants are at lower energy level, but the ball that is placed on the hill has to overcome a little hump, as you can see on the slide behind me. This ball has to be provided small amount of energy in order to go over the hump. That is the activation energy. Our products are at a specific energy level, the reactants are at a specific energy level, and the products are at a lower level. However, the reactants have to go to a transition state level, which is even higher energy level yet. In order for reactants to go to higher energy level of transition state, they require little energy. For example, combustion of methane is a very exothermic reaction. It releases a lot of energy. But as you have seen, when you turn on your stove, you have to provide some energy. You have to light up a match in order for your stove to start the combustion process. So the methane gas, which is coming out of your stove, it starts to burn. So that amount of energy, different chemical reactions have different amounts of activation energy requirement. Enzymes can lower the activation energy requirement of a reaction. Let's look at how enzymes, we will talk about how enzymes lower the activation energy requirement, but let's look at a process with and without enzymes. So at normal temperatures, only few molecules have enough energy, kinetic energy. Kinetic energy, I would like to mention, is basically the average kinetic energy of the system. That is the definition of temperature. So at a specific temperature, say for example 25 degrees Celsius, we are saying that the average kinetic energy of molecules of methane, for example, is a specific number. However, this is an average. Certain molecules will have higher energy, higher kinetic energy. Other molecules will have lower kinetic energy. The molecules that have a higher kinetic energy can overcome that activation energy barrier and can get converted into products. So the process is very, very slow, as we looked at an example of degradation of polyarginine. Enzymes can help the products attain the transition state with a lower energy requirement. So as you remember, we also saw the case of glucose 1-phosphate and converting into glucose 6-phosphate. The question was, why is it that 5% of the molecules are still at a higher energy level? We said that that process is occurring at 25 degrees Celsius. At 25 degrees Celsius, some molecules had enough kinetic energy and they could use that kinetic energy to attain a higher energy state, which was glucose 1-phosphate. So their kinetic energy was converting into potential energy in the form of high energy bond, which was glucose 1-phosphate bond. Enzymes can use 3 different techniques, 3 different strategies to lower the activation energy. We'll look at that in the next module.